Thank you for the invite. I'm very happy to be here. So today I wanted to talk to you about some of the work and infrastructure that we've been building at Cohen Veterans Bioscience to help fast track the discovery and development of biomarkers for post-traumatic stress disorder and traumatic brain injury. We are a nonprofit research, biomedical research organization, and in addition to wanting to accelerate the discovery and development of these biomarkers, we act as an integrator for many different stakeholders in this area. We are able to work with nonprofits like ourselves, but also government, academia, industry, and then also globally as well. So we're not limited to the US. In addition, we do really want to push the idea of a mechanism-based approach in terms of characterizing psychiatric disorders. Right now, it's, of course, symptom-based subjective assessments that are done, and partly because we're not understanding much about the biology, but we want to really push the evidence-based approach. Further, this is something I've been talking about with a number of people, is that we want to also look into transdiagnostic approaches. With neuropsychiatric disorders, there's a lot of overlap in between the symptomology. You see things like anxiety, depression, trouble sleeping across many different disorders. And so there may be opportunity to identify the biological mechanisms that drive these and be able to treat these components effectively. So here's some of the reasons why we are focused on these disorders. So in terms of the diagnostics and potential treatments for these disorders, it's a little bit barren here. For PTSD, there are no diagnostics. For traumatic brain injury, there are, I think this actually might be 7 aids in diagnosis. So nothing that can be done as a standalone, but as something in addition. Unfortunately, they're not normally done. So not utilized in the context of use that they were developed. In terms of treatments, so nothing for TBI at this time. And in PTSD, we have 2 SSRIs that were approved about 20 years ago and are not very effective. And then we have one that's actually new, which is for nightmares. And so it's an app that you can put on your Apple Watch. And if you're having a nightmare, it can sense that's coming on and it'll wake you up. So it's not treating the disorder, but it's inhibiting the symptoms from happening. So that's what we have. So we have some work to do in this area. So we want to take what has been learned from all of the indications that have been being discussed today, throughout the day, and really drive home the ability to harness the use of these emerging technologies, the cutting edge analytics approaches, and really get a better idea of what's going on in PTSD and TBI. We do know there's a lot of heterogeneity in these disorders. We see, very different outcomes, very different symptom clusters. And so, really understanding potential biotypes is really important. And so we really need to dig deep into, these very well characterized participants, very well standardized collected samples to understand, what is going on in these disorders. And so we want to look at the multiple levels that have been, being discussed today. And we think that this is really the only way we can get to effective treatments. All right, so I'm going to talk to you today, as I mentioned, about the infrastructure we've been building at Cohen Veterans Bioscience. And I'm going to take you through a super brief tour of how we are using it to really, drive the discovery, but also pressure test certain biomarkers for PTSD specifically. And I do want to mention that I will be focusing on plasma today. We don't have brain samples from individuals with PTSD. So this begins with our foundation, which is the biorepository. We sponsored a number of cohorts, and these were all very well characterized. We're continuing actually to sponsor a number of other cohorts, but making sure we understand a lot about these individuals. Things like medication and co-occurring disorders are really imperative to know if you want to understand the relevance of certain biomarkers, as I'm sure we all can appreciate. In addition, we employed standard operating procedures for the collection and processing of the samples. Next, we developed a biofluid assay evaluation paradigm. As I'm sure everyone is well aware, not all assays are created equal, and we needed to understand which ones were best for these types of samples, for this population, for these types of co-occurring conditions. So we developed a paradigm that I'll go through today. Third, we have our own internal data science team. And so this is something that is actually quite unique to our organization. I'm able to work hand in hand with a number of data scientists, individuals that have expertise in imaging, EEG, and all the multi-omics approaches. And so we really get to work together to address the important biological and clinical questions. And then lastly, we have our BRAINCommons, which was actually mentioned, I believe, in the first session, which is a data sharing platform, which is really useful for working with partners to use new approaches with this data and be able to access the data efficiently. So let's take our tour. So the first thing is, so for PTSD, there have been biomarkers that have been reported. So I like this slide because it shows all of these different biomarkers that we think might be important. And, they kind of make sense if we think about, hyperarousal or stress response. But there's a little bit of we don't have a lot of clarity about what they mean. And I'll go further into that in a moment. So our case study that we're going to go through today is going to go through inflammatory biomarkers. And these have been shown in many reports. There's some kind of component in PTSD that we're getting an inflammatory response. What that looks like is highly disputable. Now, our overall goal was to understand if we could use inflammatory biomarkers as a potential diagnostic for trauma-related brain disorders. But we then also had the access to 2 different cohorts, which are longitudinal. And so in a unique study, we're looking at how these the inflammatory biomarkers might change over time in response to treatment. And so is this something that could be treated? So this slide is demonstrating some of the challenges that we have. So on the bottom, we have some of the most commonly reported biomarkers for PTSD. And then on the y-axis, you have the number of reports that have talked about these biomarkers. For example, BDNF, which is this one, mechanistically, this makes sense, this would be important. You have four reports of it going up in the plasma, you have 5 of it going down, and then you've got, I believe, about 8 where it doesn't do anything. And so this is how it is. This is how the literature is. There's no consensus one way or another for any of these biomarkers that have been measured. But there's, none of these are wrong, is the thing. We know that people use different assay methods. Some of them, for example, for cytokines, they're very low levels in the plasma. So perhaps the assay wasn't sensitive enough. But I think more importantly is that there are variable clinical study conditions. And so for the cohorts I'm going to speak about today, these are 2 cohorts of veterans. They're from OIF and OEF, so Operation Iraqi Freedom and Operation Enduring Freedom. So very specific age, also mostly male and largely white. So it's a very homogeneous population. Now there's all these other studies are, they come in many different flavors because trauma is not, a distinct event for just veterans. It's something that, takes place all over the world. And so, thinking about children, thinking about women, thinking about older individuals, these are all, they all have very different profiles when you think about what's going on biochemically. And then lastly, for a lot of the earlier studies, taking in consideration the co-occurring conditions and the medications wasn't always something that was included. And so for inflammatory cytokines, it's something that is essential for us to really understand if it's a meaningful effect. All right, so we'll start with our inflammation assay comparison. So this was a cross-platform comparison that we did in partnership with the Michael J. Fox Foundation. We looked at these 12 cytokines that were important to both organizations, and we looked at different performance metrics. These are the 5 platforms that we investigated. This was 2019 when we made these decisions. So, there may be newer versions at this point. And then we looked at these 4 cytokines on the left into a deeper analysis that we reported in the literature. And with this, we were able to create a scorecard to understand, which assay, which platform would be optimal for the samples that we had for PTSD and then also Parkinson's disease. So that's reported here. And that was the Quanterix Simoa platform. Now, the next thing is, samples. Which samples should we use? So these are the 2 cohorts that I was referring to. Both of them, like I said, are veterans, and they are longitudinal. They went through different courses of psychotherapy. So one of them was a 12-week course, which had prolonged exposure and cognitive processing therapy. And then the other was an intensive outpatient program that was only 2 weeks long. And so, again, these individuals are very well characterized. We have the ICD codes, we've got all their medications, full clinical write-up. We also have imaging, EEG, and then a number of other samples were collected as well for future analyses, because we do want to do, all the different types of omics to really understand these individuals. So taking these samples, we ran them on the Quanterix Simoa, and I just want to give you just some quality control. Like, here's what the data looks like, what we found. And we were actually quite happy with the results. So this is in plasma, IL-6. We used equivalency controls to stimulate the cytokines and make sure that we were able to measure them in this matrix. Everything looked pretty good. So these are actually replicates that you're seeing here, and most of them are quite precise. So we were happy with this data. Here's another example of IL-1 beta. This is a notoriously low cytokine. You're not going to have it circulating normally. It's something that is only happening when you're sick or you're injured. So we weren't actually surprised to see most of these are under the level of quantification. So happy with these results. But one thing I wanted to share with the group today is that we had implemented the standard operating procedures for the collection and processing, and we looked across the 2 cohorts at these 6 cytokines here, and we do see alignment. Now, these are different sites, different PIs, different states. So and cytokines, they're pretty finicky, and so we were very happy to see that we did see quite a bit of alignment, and this is at baseline before psychotherapy. So we're not comparing those different approaches, and so standardizing that collection may allow for more meaningful comparisons. We do think that for broader use of other cohorts, using things like the genetics and epigenetic approaches probably will be more robust in terms of stability, but the fact that we could implement this was very positive. So just our initial analysis, was that the Simoa platform worked well for these cytokines, and that our standard operating procedures were effective to allow consistent measurements across the different sites, and that having the information about potential impacting factors is really important. All right, so our next steps are to do the good stuff, the fun stuff. We're going to look at what is the changes that we see longitudinally. Are there differences between responders and non-responders? We do think that there may be, a discrete group that is, they have inflammation that is driving the disorder. So this is something we're very interested in looking at. And so we'll be able to really understand when we look at the different clinical outcomes. And then we also want to combine these and do a multi-modal approach and perhaps develop some predictive models. So our takeaway is that we've established this critical infrastructure to really drive the discovery and development. And we're continuing to do this with other omics right now, not just the cytokines. That was just a little taste. We're continuing to do this and working through that. Using this approach, we do think we'll have a lot more confidence in the results and really pressure test these different biomarkers. And so let us understand what is the utility and maybe the lack of utility for them in certain contexts. And then we are encouraging data sharing by our BRAINCommons platform and following best practices, of course. And I did want to mention that we love partnering. We're a small organization, and hearing about all these new approaches, new ways to look at these different types of data has is very inspiring. And so if we're able to work with someone with a new approach, a new idea, we'd love to do that. So if you'd like to come with us on this journey, please reach out. And that's all. Thank you.